Okay, today we're going to discuss about the antimicrobial activity. Okay, so this topic will be discussing about the different actions of our antimicrobial agents that would enable them to kill your bacteria. So these are actually um, mechanism of your antibiotics here, antibiotics for the way and how they were able to kill your bacteria or your pathogen. Okay, so we have the term antibiotics. So antibiotics here are substances that has been obtained and purified out of your antimicrobial agents. So it may came from your bacteria or may came from here the fungi or even from other or even your plant extract that would kill your bacteria. So antibiotic would only be effective against your bacteria. It's not for the viral infection. Okay, we have the term bacteriostatic versus your bactericidal. So when we speak about bacteriostatic is just suppress the bacterial multiplication or their cellular division that would inhibit them to multiply para hindi na sila dumami. When speak about bactericidal, okay, is eventually the killing of your bacteria. Okay, now we go to the different actions of our antimicrobial agent. The first action of your antimicrobial agent here, it try to inhibit the cell wall synthesis. So remember here that the cell wall of your gram-positive, the gram-negative is made up of your peptidoglycan. Only that your gram-positive would have a thicker peptidoglycan layer. For the gram-negative, so it's made up of two layers, diba? The inner membrane of your gram-negative is made up of the thin peptidoglycan, but the outer membrane is most likely made up of your lipids, the LPS, our lipopolysaccharide. Okay, so antibiotic here that try to inhibit your cell wall synthesis or simply inhibiting the synthesis of the peptidoglycan. Okay, the cell wall of your bacteria try to protect your bacteria against the osmotic pressure. So if the bacteria could not synthesize the cell wall or the peptidoglycan, it eventually result here to the lysis due to your osmotic pressure. Hindi nila kaya ang osmotic pressure. And they die by lysis because, of course, they don't have here the peptidoglycan. If there will be problem with the synthesis of your cell wall or your peptidoglycan. So again, going back here to the basic structure of our peptidoglycan. Okay, so peptidoglycan is made up of your polysaccharide. This is a sugar with a repeating unit of your NAM and acetyl muramine. And we have also your NAG and acetyl glucosamine. Okay, so if this one is one polysaccharide layer, so we have your NAG, NAM, NAM, NAG. And then again, at the end of your NAM, so there are 4 amino acid attached to that one, or you call that one a tetrapeptide. So we have your L-alanine, we have also here your D-glutamate, mesodiaminopymylate, and we have your terminal D-alanine. And then we have also here another polysaccharide. So again, made up of the NAG, the NAM, and the NAG, NAM ulit. In the NAM again here, there's a 4 tetrapeptides which try to attach to the NAM. Okay, this includes still again your L-alanine. Okay, we have your mesodiaminopymylate, D-glutamate, and the terminal, ang pinaka-end part is the D-alanine. Okay, so each of this polysaccharide unit is eventually being linked or cross-linked by the enzyme. So we have your transpeptidase. Transpeptidase try to link here your terminal na D-alanine. D-alanine here from one polypeptide, and then we have your D-alanine of another polypeptide. Since they are proteins, so they are cross-linked by the transpeptidase that would allow them to be cross-linked or to attach to one another. On the other hand, each of your sugar, the glycan, okay, they are being linked here to one another by the enzyme transglycolase. So in order for you to have a bigger structure, okay, so pag ganyan, okay, so one polypeptide, when we have another one, polysaccharide, other one, so there are cross-link here. In order for you to make here a very complex na peptidoglycan layer. Okay, we have here the steps for your synthesis of your peptidoglycan. For the first one, we have here the synthesis of the precursors. Precursors are the substances which are needed in order for you to make your peptidoglycan. So first, you need to have your polysaccharide, your NAM, the NAM and NAG. NAM and NAG. Then we have also here your tetrapeptides. Okay, after that, after you have that precursor, this one will be, if this one is your cell, okay, so basically this precursor, they are synthesized outside the body of your bacteria. Okay, then after that one, or they're in the plasma, or this is your bacterial cells. So after that one, after they are synthesized here outside, they will be transported here paloob. Okay, that's transport to the cell membrane. So since they are part of your cell membrane, ang ating peptidoglycan, your cell wall. And after that one, insertion of your glycan. Okay, so if you have already here, nakaumpisa na siya paggawa, so para dumami siya, mag-insert ka ng another layer of this one. And lastly would be the cross-linking by the action of your enzyme transpeptidase and transglycolase. Transglycolase. Transpeptidase, transglycosylase. Okay, that would eventually produce here a very complex structure, cross-link structure of your peptidoglycan. Okay, now we go to the different antibiotics here that try to inhibit your cell wall synthesis. So, first we have here the beta-lactams. So, the beta-lactams contain here the beta-lactam ring. The beta-lactam ring resemble here the structure of the precursor na D-alanine tetra. We have here pentapeptidyl glycosylase. So, parang pareho nito ang structure ng ating beta-lactam ring. So, ito lahat. Okay, so ito. Okay, so since pareho sila ng structure, pag pumasok dito sa beta-lactam ring natin, your bacteria could not, this one, pag antibiotic dito, hindi siya makapag-insert or hindi ka makapag-cross-link. Okay, so therefore, hindi maproproduce ang ating peptidoglycan. Okay, the target of your beta-lactam ring here would be your PBG or your penicillin binding protein. Penicillin binding protein are actually the enzyme. This is the bifunctional enzyme. Ito yung enzyme na transpeptidase saka transglycosylase. Yung transpeptidase, ano ginagawa ni transpeptidase? Again, it try to cross-link our amino acid. Okay, in order for that to be incorporated or to cross-link here between your dalawa na polysaccharide unit. Okay, especially the NAM and the NAG, the NAM, dalawang terminal na NAM, for them to be cross-linked to one another. Another one, this one is another enzyme, transglycosylase, responsible here for cross-linking between your polysaccharide units. Okay, so basically the target of your beta-lactams will be the enzyme. Okay, so that's the enzyme needed here for the transpeptidation or the transglycosylation or the cross-linking in order for you to have here a highly complex na peptidoglycan layer. Okay, so first we have your penicillin and then your cephalosporin. So these are the antibiotic here that try to inhibit the maturation of your enzyme. Na PBG, this is your enzyme. This is your protein na penicillin binding protein, but this refer to your enzyme. So once na hindi mag-mature ang ating enzyme, they will not involve here. It will not allow the transpeptidation and transglycosylation. And therefore, hindi ka makapag-produce na highly complex na peptidoglycan layer. Okay, another one, we have here the group of your penim, sepim, carbapenims, monobactams. On the other hand, try to inhibit here the enzyme itself. Okay, so even if you have your enzyme, mature enzyme, but try to inhibit the activity of that. Okay, try to inhibit here your PBG, which are the bifunctional enzyme. And therefore, also try to inhibit pa din ang ating cross-linking between your peptidoglycan layer. What's going to happen here, you have just a linear na peptidoglycan. Pag sabing linear, so ito lang, ganyan. Or a non-synthesis of that. Wala ka talagang masynthesize na peptidoglycan layer. Another group of your antibiotic here that try to inhibit your cell wall synthesis, we have your glycopeptides. The glycopeptides, this includes your vancomycin, dalbavancin. Okay, that try to inhibit here the precursor. Dito mismo sila sa precursor na PBG. Sorry, nabura. Okay, so glycopeptide, on the other hand, they try to inhibit here the synthesis of the precursor substance. So, itong precursor substance natin, itong kailangan natin para makapag-produce ka. So, pag hindi sila na-synthesize, eventually, you could not able to form also here your peptidoglycan layer. Okay, so this includes your vancomycin. Vancomycin is only effective for the gram-positive. This is naturally resistant to your gram-negative. Hindi talaga maging effective ito. Then dalbavancin as well. So your beta-lactams, sa enzyme sila, ito naman sa precursor natin. Okay, so ito ay mismo sa enzyme. This one ay the precursor of your enzyme. Enzyme na PBG. This one is the precursor na PBG. Ito. Ito yung precursor natin. Ang enzyme natin dito includes your ito. Ito ang ini-inhibit ng ating mga beta-lactams. And all this one result to your non-synthesis of your peptidoglycan layer or the cell wall. And therefore, pag wala silang cell wall, they die because of the osmotic pressure. Okay, the next group of antibiotic here try to kill the bacteria by inhibiting the cell membrane function. So basically, the cell membrane, the bacteria try to function as number one is the site of your electron transport chain. Electron transport chain would provide here the necessary ATP or the energy for the cellular respiration of your bacteria. Another one, also try to regulate here the osmotic pressure. And if the cell membrane function here has been disrupted, that would eventually result here to the cell death. So the first one, we have here your lipopeptide. So this includes your lipopeptide. So we have here your daptomycin. So daptomycin specifically try to inhibit here the cell membrane function of your gram-positive na bacteria. Then we have also here your polymyxin. So we have polymyxin B and colistin. So this is a type of your lipopeptide that try to inhibit here the cell function of your gram-negative na bacteria. Okay, the next one, we have here antibiotic which try to inhibit here the protein synthesis. Basically, they try to inhibit here the translation process. Okay, so when we're having the translation process, so as a review, we have here the steps for the translation. What you are needing here for the translation, we have the messenger RNA. Messenger RNA contains here the triplet of your base, your amino acid, three amino acid, or three nucleotide base. Okay, so this one is could be found here in your messenger RNA. Okay, then what you'll be needing also here would be your ribosomal RNA. So there are two ribosomal RNA here. We have yours, okay, small subunit, the 30S, and we have here the big subunit, we have here the 50S. Okay, another one, you'll be needing also here the transfer RNA. Transfer RNA, try to carry the anticodon, which is anti-complementary here to your codon. Okay, so they just have here the base pairing for that. So, again, ganyan. Okay, so anticodon mo is your 80, diba? So, this one is A, then what is C? Okay, this is your, if this one is your codon, we have also here your anticodon for that. And that is being provided here by your transfer RNA. This one trying to carry your anticodon. And this anticodon here will bind with your messenger RNA through your 30S RNA when they form a complex by this enzyme, amino acyl transfer RNA synthetase. Again, this is the enzyme that would allow the binding of the transfer RNA to your complex mRNA 30S and then the 50S. So pag nag-bind na siya dito, it will be translated. So start with your start codon. Okay, then it will stop with the stop codon. So, pag nag-translate na siya, so dito mo na siya, ang incoming na mga anticodon natin, they will able to bind here with your ribosomal RNA through SA site. Then, try to translate. Pag na-translate na siya, ang ating mga 3, yung codon natin magiging amino acid. Like in the case of your AUG, this is your codon. Okay, magiging ano ito, magiging methionine. So, pag after translation, so magbabind ito dito, ita-translate niya. Pag na-translate, idadali niya dito sa P site. So, what you see here would be, the P site would be your translated na mga amino acid. Okay, then, next naman, magdadala ulit sa anticodon. Ita-translate ulit from A site, dadalhin sa P site. Okay, so that become your incoming amino acid. Okay, so the newly synthesized na amino acid or the incoming amino acid will bind to the existing na amino acid. Mas sabing existing, ito yung una mo na-translate. So magbabind siya dito by the enzyme peptidyl transferase. Okay, so that would eventually allows it na mag-elongate. So, para dumami ka. Pag dumami ng mga amino acid mo, okay, it will assemble that one to become a structural protein. Okay, so that you have your protein. That's the entire process of your translation. Okay, now we go to the different antibiotic here that try to inhibit on this process, your protein synthesis. So the first, we have here the antibiotic will try to inhibit here your 30S, ribosomal RNA. The first one, we have your aminoglycoside. Aminoglycoside includes here the group of your antibiotic, which includes your gentamicin, amikacin, and we have also your tobramycin. So this antibiotic here try to inhibit your 30S ribosomal RNA by inhibiting here the docking of your amino acid transfer RNA to your messenger RNA. So ito yung mga antibiotic natin na, okay, so dapat ito, diba, yung transfer RNA with its amino acid transfer RNA synthetase. So mag-a-add siya ng anticodon dito. Idadali niya dito dapat. Ang ginagawa ni aminoglycoside, they prevent that one na mag-bind dito sa ating P site of your ribosomal 30S ribosomal RNA. And therefore, pag hindi siya makapag-bind, it will not translate. Wala kang protein na magagawa dito. Another one, we have your tetracycline. Tetracycline try to block here the elongation process. Paano siya ibablock ang elongation process? So basically, di ba, para mag-elongate itong ating amino acid na mga na-synthesize from this area, dadali mo dito, okay, sa ganitong area. Okay, and eventually, that's would okay, from this area, dadali mo dito sa area na to, and that would eventually result here to the elongation process. Pero sa tetracycline, dito lang siya, it will not allow it papunta dito. And therefore, preventing here your amino acid, translated amino acid, to be transported to the P site, and therefore prevent here the elongation process. Wait, from the A site, okay, to your P site. Ay, wait lang. Check ko. Okay, so, naguluhan ako kung alin ang A site sa P site natin. Okay, tama. We have A site, tapos P site. Okay, so ang ginagawa ni tetracycline, prevent here the transfer of your amino acid from the A site papunta dito sa P site. Pag hindi siya pumunta dito, it would not allow it to elongate. Okay, your glycine on the other hand, so this is it's just a derivatives of your tetracycline, only for those na patient which are resistant to your tetracycline. So, same activity ni ating tetracycline, prevent here the chain elongation by preventing your newly synthesized amino acid from your A site to be transferred to your P site, and therefore preventing also the chain elongation. Okay, now we go to your antibiotics which try to inhibit here the 50S ribosomal RNA. So we have the group of your MLS, macrolide. Macrolide includes here your erythromycin, azithromycin, and we have also here your clarithromycin. Then we have your L, stand for the lincosamide, and S for the streptogramine. streptogramin. Okay, so this MLS here try also to prevent the chain elongation basically by blocking the exit tunnel in the A site. Okay, so hindi makalabas ang ating amino acid on this area. In your P site, I mean. Okay, try to block yung exit tunnel here. So therefore, pag na-block ito, it will eventually not able to elongate then. Okay, so that would result here to the premature release of your amino acid and therefore preventing also here the chain elongation process. And we have also here antibiotics. We try to also still inhibit here the RNA synthesis by inhibiting basically the 50S ribosomal RNA. So we have here your oxazolidinones. Example for that, we have here your linezolid. So this antibiotic here try to inhibit the protein synthesis by inhibiting the pre-initiation step. Pre-initiation step here is the first step primarily in your protein synthesis. So it involves here the binding of the entire complex 30S messenger RNA and a 50S ribosomal RNA. So if the pre-initiation step will not occur, so hindi sila magbabind, hindi sila magpa-form ng complex ang tatlo na to. Ito. And therefore, saka ito. And therefore, hindi tayo makapagkaroon, hindi tayo makapag-translate ng ating amino acid into your, and would have here result also to preventing your protein synthesis. Okay, another one, we have here your chloramphenicol. We try to inhibit here the chain elongation, basically by inhibiting the enzyme peptidyl transferase. Okay, your peptidyl transferase is the one we try to transfer your newly synthesized na amino acid or incoming amino acid to your existing na amino acid in order for you to have a long chain of that. So pag wala kang nitong enzyme, it will not allow here the chain elongation and eventually wala kang protein, complex protein structure na mapoproduce. Okay, next we have here another antibiotic we try to inhibit here or try to kill bacteria by inhibiting your DNA synthesis and your RNA synthesis. So this group of antibiotic try to inhibit basically your DNA replication and your transcription process. So kanina is your translation process. And this one naman that involves here your replication and even your transcription. First one, we have your fluoroquinolones. The fluoroquinolones here, example, your ciprofloxacin. Okay, that one, try to inhibit the enzyme DNA gyrase or that's your topoisomerase. Topoisomerase help here para ma-unwind ang ating double helix. Okay, kailangan ng topoisomerase para ganyan to prevent the reformation of your double-stranded helical na DNA in order for them na mag-separate ang dalawang strand, the leading and your lagging strand, in order for you to copy by the process of your DNA replication. So, pag walang mag-unwind ng ating double-stranded DNA, it will not able to copy that one by the process of your DNA replication. And therefore, wala ka ding translation. Okay, di ba? Start with the replication, followed by your transcription, and we have your translation. Kaya pag affected dito, it will affect also the protein synthesis. Your metronidazole, on the other hand, so basically, you have already an unwind na leading strand sa lagging strand. However, it try to break your strand. Okay, so pag it try to break your leading and the lagging strand, wala din siyang ikakopy. Okay, that would still inhibit your DNA replication. Another one, we have your rifampicin. So this is effective for your tuberculosis. And it try to kill your Mycobacterium tuberculosis by inhibiting the transcription process. So this antibiotic basically try to inhibit the activity of your RNA polymerase. Okay, your RNA polymerase try to be responsible for transcribing your DNA to your messenger RNA. So, ang DNA natin, it will be transcribed to your messenger RNA by your RNA polymerase. And this antibiotic rifampin here would try to inhibit the action of your RNA polymerase, preventing your transcription process. Okay, then we have here the group of your antibiotics to try to inhibit your folic acid synthesis. So we have here the step in your folic acid synthesis. We start with your PABA or para-amino benzoic acid converted to your dihydropteroate by the enzyme dihydropteroate synthetase. Then we have also here dihydropteroate converted to your dihydrofolate with the then we have also dihydrofolate is further catabolized to your tetrahydrofolate with the enzyme of your dihydrofolate reductase. Converting that one to your folinic acid purines, and we have also here your DNA, folic acid. Okay, then we have your antibiotics which try to inhibit your folic acid synthesis. We start with the sulfonamides or your sulfamethoxazole. Ito yung mga sulfa drugs natin. So basically, this antibiotic here try to inhibit this enzyme, dihydropteroate synthetase. Nakalagay sa PowerPoint nyo, dihydropteroate reductase, but that one is your dihydropteroate synthetase. So kindly make correction on that. Then next, we have your trimethoprim. Trimethoprim here would try to inhibit this enzyme, dihydrofolate reductase. Okay, basically your sulfamethoxazole and trimethoprim, they try to exist in a synthetic na antibiotic in the form of your cotrimoxazole. So, cotri, your combination of your sulfamethoxazole and trimethoprim. Then we have also here your nitrofurantoin. So, this one is used for the treatment of your urinary tract infection that try to inhibit your bacterial protein and your enzyme synthesis.